A recent study published in Aging Cell delves into the intricate challenges of restoring hair growth through stem cell-based therapies. Researchers have identified a promising approach that addresses the pervasive issue of cellular senescence in cultured cells, a significant hurdle in the development of effective hair regrowth treatments. This advancement revolves around the use of senolytics, a class of compounds designed to selectively eliminate senescent cells. These cells, which cease dividing and release inflammatory molecules, accumulate in cultured dermal papilla cells, hindering their ability to promote hair growth. The study's findings mark a crucial step forward in overcoming the limitations that have plagued regenerative hair restoration research for years. At the heart of the study lies a challenge that has stymied researchers for decades: maintaining the functionality of dermal papilla cells outside the natural environment of hair follicles. These mesenchymal stem cells are central to the hair growth cycle, but when isolated and cultured, they quickly lose their hair-growing potential. Unlike rodent cells, which exhibit greater resilience during repeated passages in culture, human dermal papilla cells experience a rapid decline in potency, rendering them ineffective after a few passages. To date, various strategies have been employed to restore the functionality of these cells. Approaches such as using conditioned media or embedding cells into extracellular matrices have shown some promise, but their efficacy has been largely limited to cells in the early stages of culture. As cells age in vitro, their ability to promote hair follicle formation diminishes, presenting a significant barrier to clinical applications. The decline in dermal papilla cell functionality is closely tied to the phenomenon of cellular senescence. In cell culture, senescence arises when cells lose their ability to divide and accumulate damage, leading to the secretion of pro-inflammatory molecules collectively known as the senescence-associated secretory phenotype, SASP. This inflammatory microenvironment not only impairs the senescent cells themselves but also negatively influences surrounding healthy cells, further compounding the problem. The study demonstrated that dermal papilla cells are particularly prone to senescence compared to other cell types, such as dermal fibroblasts. From the very first cellular passage, these cells exhibited hallmarks of senescence, including the activation of specific biomarkers and a reduction in proliferation. As senescence set in, the release of SASP factors such as interleukin-6 and interleukin-8 created a self-perpetuating cycle of dysfunction. Unlike in living organisms, where the immune system plays a role in clearing senescent cells, such mechanisms are absent in cell cultures. This allows senescent cells to accumulate unchecked, creating a toxic environment that undermines the effectiveness of cultured dermal papilla cells. To counteract the detrimental effects of senescence, the researchers turned to senolytics, compounds that selectively target and eliminate senescent cells. In living organisms, the use of senolytics is controversial due to the complex roles that senescence plays in processes like wound healing and cancer suppression. However, these concerns are irrelevant in the controlled environment of cell culture, making senolytics a compelling solution for this application. The study explored the impact of senolytic treatment on cultured dermal papilla cells. When senolytics were administered during the third passage of cell culture, they effectively eliminated senescent cells. Importantly, many of the remaining cells entered a quiescent state, characterized by reduced activity but preserved potential. Quiescent cells can be reactivated to fulfill their functions, unlike senescent cells, which are irreversibly damaged. The elimination of senescent cells also led to a marked reduction in SASP factors and their downstream effects. By restoring a healthier microenvironment, the senolytics enhanced the overall viability and functionality of the cultured dermal papilla cells. To validate the effectiveness of senolytic-treated cells, researchers conducted a series of experiments involving both animal models and human skin cultures. In one experiment, treated dermal papilla cells were used to create spheroids containing a mix of human dermal papilla cells and mouse keratinocytes. These spheroids were implanted into the backs of hairless mice, resulting in significantly improved hair growth compared to control groups that did not receive senolytic treatment. A similar experiment was conducted using cultured human skin, where treated cells were embedded in collagen matrices. The senolytic-treated cells demonstrated the ability to grow keratin in hair follicle structures, whereas untreated cells failed to do so. These findings underscore the transformative potential of senolytics in revitalizing dermal papilla cells and enhancing their capacity for hair follicle regeneration. While the results are promising, the study highlights several limitations and areas for further exploration. One key observation is that the effectiveness of senolytics diminishes with later passages of cultured cells. Cells treated during earlier passages showed a much greater capacity for hair growth than those treated later in the culture process. This suggests that while senolytics can mitigate the effects of senescence, they cannot fully reverse the damage in heavily aged cell populations. Another challenge lies in the translation of these findings to practical therapies for human hair loss. The experiments, while successful, were conducted in controlled laboratory settings. Developing a treatment that replicates these results in clinical settings will require advances in the engineering of artificial human skin capable of supporting hair follicle growth. Moreover, the study raises important questions about the long-term viability of senolytic-treated cells. While the immediate effects on hair growth are encouraging, it remains to be seen whether these cells can sustain their functionality over extended periods or whether additional interventions will be necessary to maintain their potency. The research represents a significant milestone in the quest to develop effective hair restoration therapies. By addressing the root causes of cellular dysfunction through the use of senolytics, scientists have opened new avenues for overcoming the challenges associated with culturing dermal papilla cells. Future studies are likely to focus on optimizing the timing and dosage of senolytic treatments to maximize their efficacy. Additionally, efforts to integrate these findings into more complex tissue engineering approaches could pave the way for creating bioengineered skin with fully functional hair follicles. While a readily available treatment for hair loss remains on the horizon, the insights gained from this study provide a strong foundation for further research. By harnessing the power of senolytics and other emerging technologies, scientists are bringing us closer to a future where hair loss can be effectively treated using regenerative medicine. This breakthrough underscores the transformative potential of targeting cellular senescence, not only in the context of hair restoration but also in broader applications of tissue regeneration and aging research. As the field continues to advance, the lessons learned from this study could have far-reaching implications for improving human health and combating age-related decline. Thanks for watching. If you found this video insightful, don't forget to like, share, and subscribe for more updates on cutting-edge science and health breakthroughs. Let us know your thoughts about this exciting research in the comments below. Would you try a senolytic-based hair restoration treatment? Stay tuned for more content, and until next time, stay curious and take care.